2. Introduction. A population is a group of individuals of same or closely related species living in the same area at a given time and has no barriers to exchange of genetic material. For example, population of human beings living in a city, population of lions living in a forest, etc. The study of structure and dynamics of population and their interactions with environment is known as population ecology. The study of population ecology plays an important role in protecting and managing the rare species through various means including population viability analysis (PVA). PVA helps to determine whether a population would survive or it would face the risk of extinction under the present environmental conditions. Further, each population has a minimum viable size, the size at which it can avoid the extinction due to various biotic and abiotic factors. The study of principle of inheritance of characters to large group of individuals in a population is called as population genetics. It can be used to examine the patterns of genetic variation within the group of individuals. In other words, population genetics can be used to study the pattern of genetic variation in a populations and how it changes in different area and at different time. The principal aim of population genetics is to understand the evolution in terms of genes, which occurs in a Mendelian population or a species over a period of time. 3. Population genetics also play a key role in understanding the value of genetic resources and its use in conserving biodiversity. 3. Unitary and modular population. The individuals form the constituent of a population. The individuals can either be distinguished as single unit, like dogs, cats, etc., or those organisms which do not distinguished as simple unit like grass, etc. On the basis of their distinguished character, population can be divided into two types: 1. Unitary populations and 2. Modular populations. In unitary populations, each organism is resulting from a zygote whose growth is determinate and predictable. These individuals can be distinguished as single unit like dogs, cats, spiders, insects, fish, birds and amphibians. However, in case of modular population, an organism develops from a zygote and serves as a unit module from which several other modules are produced, forming a branching pattern such as plants, sponges, hydroids, fungi, bacteria and corals. The structure and pattern of modular organisms is indeterminate and unpredictable. Many plants, grasses and perennial herbaceous plants grow root extensions to form new shoots or suckers that may remain attached to root extensions or break off to live independently. These new individuals grow and cover a considerable larger area but appear to be an individual. The individual tree or plant produced by sexual reproduction and thus arising from a zygote is a genetic individual or genet, but modules produced asexually by the genet or ramets. In a grass or weeds, a number of young plants arise through runners or stolons. When they separate from the parent, form new plants. These groups of new plant are called as ramets. The ramets either remains attached to the parent genet or they may separate and grows independently into new individual. Individuals of ramets are genetically same and replica of parent plant. A group of ramets which develop from a genet is called as clone because all of them are genetically alike. The formation of ramets in the plant is common and can be easily seen in grasses. The populations can be R-selected or K-selected based on their strategy of survival and growth. The R-selected populations are characterized by high reproductive rates, rapid utilization of resources over a short period of time and high rates of intrinsic rate of increase. 4. R. These populations exhibit shorter lifespans and are opportunistic. On the other hand, K-selected populations are slow-growing and their numbers are limited by the carrying capacity of the environment denoted by K. These populations show higher survival, high competitive ability and low rates of intrinsic rate of increase. R. 4. Gene pool and gene frequency. The sum total of genes of all the individuals of a Mendelian population constitutes gene pool. It explains the proportion of different kind of genes distributed among the individuals present in the population. For example, the gene for polydactyly in Mendelian human population is dominant, yet the polydactylous phenotype is rare among infants. It indicates that frequency of the dominant allele in lower than its recessive allele in a population, and both alleles do not exist in population in the 1:1 ratio. 4.1 Gene frequency. The gene frequency refers to the proportion of an allele in the gene pool as compared with other alleles at the same locus irrespective of their distribution in organisms. A gene frequency always ranges between 0 and 1. If 43% of the people in a group have brown hair, the frequency of brown hair in the group is 0. A gene frequency always ranges between 0 and 1. If 43% of the people in a group have brown hair, the frequency of brown hair in the group is 0.43, and the frequency of people who do not have brown hair is 10.43 equals 0.57 or 57%. To calculate the genotype frequencies at a specific locus, the number of individuals is counted. Having one particular genotype is counted and then it is divided by the total number of individuals in the population. The sum of all genotype frequencies should be equal to 1. 4.2 Allele frequencies. The use of allele frequencies offers several advantages over the genotypic frequencies. First, in sexually reproducing organisms, genotypes separate as alleles during gametogenesis and thesis alleles are passed from one generation to another generation. Consequently, only alleles have continuity over time, and the gene pool evolves when allele frequencies change. 5. Allele frequencies can be calculated directly from the numbers of genotypes. In this method, we count the number of alleles of one type at a particular locus and divide it by the total number of alleles at that locus in the population. This method is called gene counting and works for a wide variety of cases, including X-linked genes and mitochondrial genes. The gene counting method is a formula for a nuclear gene with two alleles can be expressed as follow. Number of copies of a given allele. Allele frequency equals sum of count of all alleles in the population. The percentages of gametes for a pair of alleles A and A in the gene pool depend upon the genotypic frequencies of parental generation whose gametes form the pool. Thus, if a population has dominant genotype A, then the frequency of dominant alleles in the gene pool will be relatively high and the percentage of gametes bearing the recessive A allele will be correspondingly low. Thus, the gene frequency is the proportion of an allele in the gene pool as compared with other allele at the same locus. Suppose there are 100 individuals in a population out of which 40 are homozygous dominant AA, 40 are heterozygous A, and 20 are homozygous recessive A for particular trait, then the frequency of allele A will be 80 plus 4200 equals 0.6. The frequency of allele A will be 40 plus 4200 equals 0.4. 5. Hardy-Weinberg law. The basic idea of population genetics was first given by G. H. Hardy, 1908, in England and W. Weinberg, 1908, in Germany, independently. Initially it was believed that alleles of a gene follow 3:1 ratio in F1 generation. But later it was found that heterozygous individuals 6 appears at higher frequencies in a given populations. Further studies on gene frequencies was studied by R. A. Fisher, J. B. S. Haldane and Sewell Wright in 1920-30. According to Hardy-Weinberg's law, the relative frequencies of different genes in a large randomly mating population would remain constant from one generation to another in the absence of mutation, gene flow and natural selection. The Hardy-Weinberg law has three important aspects. 1. The allelic frequencies of the genes located on autosomes in a population will tend to remain constant from one generation to another generation. 2. The genotypic frequencies of the population are determined in a predictable way by the allelic frequencies genotypic frequency equilibrium. 3. The equilibrium is neutral. That is, if it is perturbed, it will be re-established within one generation of random mating at the new allelic frequencies if all the other requirements are maintained. Hardy-Weinberg's law describes a theoretical situation in which a population is undergoing no evolutionary changes. It explains that if the evolutionary forces are absent and the population is large, individuals mate randomly, each parent producing roughly equal number of gametes. During mating the gametes fuses randomly and gene frequency remains constant. The genetics equilibrium of the gene is maintained and the variability in the population is also preserved. Hardy-Weinberg law depends on the following kinds of genetic equilibriums for its full attainment. 1. The population is infinitely large and the mating is a completely random phenomenon. 2. No selection force is operative. 3. The population should be closed, i.e., no immigration or emigration occurs. 4. No mutation is operative in alleles. 5. Meiosis is normal so that chance is the only factor operative in gametogenesis. The significance of Hardy-Weinberg equilibrium was not immediately appreciated. A rebirth of biometrical genetics was later brought about by R. A. Fisher, 1918, and Sewell Wright 7. 1920. Under the leadership of these mathematicians, emphasis was given on the population rather than individual or family group, which had previously occupied the attention of most Mendelian geneticists. Around 1935, T. Dobzhansky and others started to interpret and to popularize the mathematical approach for studies of genetics and evolution. 6. Genetic equilibrium. As shown by Hardy and Weinberg, alleles segregating in a population tend to establish equilibrium with reference to each other. Thus, if two alleles should occur in equal proportion in a large, isolated breeding population and neither had a selective or mutational advantage over the other, they would remain in equal proportion from generation after generation. This would be a special case because alleles in natural populations rarely occur in equal frequency. However, they may be expected to maintain their relative frequency, whatever it is, subject only to such factors as chance, natural selection, differential mutation rates or mutation pressure, meiotic drive and migration pressure, all of which alter the level of allele frequencies. A genetic equilibrium is maintained through random mating. The formula P plus Q2 equals P2 plus 2PQ plus Q2 expresses the genotypic expectations of progeny in terms of gametic or allelic frequencies of the parental gene pool and was originally formulated by a British mathematician Hardy and a German physician Weinberg, 1908, independently, and called Hardy-Weinberg law equilibrium. The Hardy-Weinberg law is important primarily because it describes the situation in which there is no evolution and thus it provides a theoretical baseline for measuring evolutionary changes. 7. Application of Hardy-Weinberg's equation. If we know the number of homozygous recessive animals in a population, which is easy as the recessive gene has expressed, then it will be easy to find out genotype frequency and gene pool of the population by Hardy-Weinberg's equation. Example 1. Let us assume that in a given rat population, 50% individuals are brown and 50% individuals are white The gene frequencies of the population will be P equals 0.58, 9, and Q equals 0.5 since the equation P plus Q equals 1.0. In this case, gene frequency as well as genotype frequency remains same. Now by using Hardy-Weinberg's equation, the genotype frequency in F1 generation will be 25% and plus 50% and plus 25% but the gene frequencies will still remain as P equals 0.5 and Q equals 0.5, it means they remain unchanged even in the next generation. In the above example, both dominant and recessive alleles are equals. However, in nature, recessive alleles are usually found very few in number. Example 2. Population of rats in a given area have 16% recessive white individuals and rest individuals are brown in color. Find out the composition of the population. If white individuals are 16%, then M2 equals 16 or Q2 equals 0.16, Q equals square root of 0.16, Q equals 0.4. After finding Q value, the value of P can be calculated by P equals 1.0 Q, or 1.0 minus 0.4 or P equals 0.6. Now to calculate the heterozygous rats, 2PQ equals 2. 0.6, 0.4 equals 0.48 or 48% of rats population is heterozygous brown and 16% were white given. So, the rest of the population is homozygous brown 148 plus 16 equals 16%. Example 3. In a 200 million population of Americans, 125,000 individuals suffer from the heritable disease, cystic fibrosis. It is a recessive autosomal disease which is expressed in individuals having homologous recessive alleles. The carriers in a population can be found out by examining the excessive sodium in perspiration and in nail clippings. The number of carriers in the population can be calculated as P2 plus 2PQ plus Q2 equals 0.5 2 plus 2. 0.5 0.5 plus 0.5 2 equals 0.25 plus 0.5 plus 0.25. Suffers equals 125,200 million equals 0.0625%. Q2 equals 0.000625, 0.0625 100. Q equals 0.025. P equals 1 Q equals 1.0 0.025 equals 0.975. P2 equals 0.975 2 equals 0.9506. P equals 1 Q equals 1.0 0.025 equals 0.975. P2 equals 0.975 2 equals 0.9506 or 95.06% are homozygous dominant individuals. 2PQ equals 2x 0.975x 0.025 equals 0.0488 or 4.88% individuals are heterozygous carriers of the disease. Q2 equals 0.025 2 equals 0.000625 or 0.0625% individuals are homozygous recessive suffering from disease. The genotype frequency indicates that out of 20 individuals, only one is acting as carrier of the disease. Example 4. In human population 1 in 20,000 is an albino. Let us find out the genotype frequency. Q2 equals 1 20,000 equals 0.00005. Q equals square root of 0.00005 equals 0.007. P equals 1.0 Q equals 1.00.007 equals 0.993 P2 equals 0.993 2 equals 0.986. 2PQ equals 2x 0.993x 0.007 equals 0.014. Although 1 in 20,000 is an albino in human population, but out of 70 individuals one is a carrier of the disease, which is 280 times high as many people are affected. Hardy-Weinberg's equation can be used to find the frequency of genotype of the individuals in a given population which do not show the symptom of disease but they act as carrier of the disease. 10. The Hardy-Weinberg equilibrium in the allele frequency pattern of a population required 3 assumptions. 1. Individuals with each genotype must be as reproductively fit as those of any other genotype in the population. 2. Population must consist of large number of individuals. 3. Random mating occurs throughout the population. The Hardy-Weinberg assumptions does not account for any change in allele frequency within the given populations. The mathematical explanation of change or dynamics in terms of allele frequencies was updated by Fisher, Wright and J.B.S. Haldane, who provided additional theoretical models and superimposed the mechanisms for change in allele frequencies upon the Hardy-Weinberg equilibrium. The changes in gene frequencies can be produced by a reduction in population size, selection, mutation, genetic drift, meiotic drive, and migration. 8.1. Natural selection. Natural selection generally determines the relative frequency of alleles in a population. It differentiates between phenotypes in a population with respect to their ability to produce offspring. One phenotype may better survive than another, one may invade new habitat more efficiently than another, or one may reproduce more efficiently than the other. Natural conditions or particular environmental situation selectively allows one organism to develop and flourish more efficiently, and one genotype is thus afforded more representation in the population's gene pool. If this selective process continues for several generations, allelic frequencies will change significantly and the potential will arise for evolutionary change. Further, the natural selection process, while acting on the total phenotype, will, in fact, influence only the heritable portion of the phenotype. If a trait has a high heritability, selection can rapidly affect its frequency within a population, whereas selection will take a great deal longer to have any effect on a trait with a low heritability. Natural selection includes the following three parameters: survival rate, relative fitness, and the selection coefficient. In natural populations, main pattern of selection is followed as directional selection. It either eliminates or reduces the reproductive potential of particular phenotypes in a population. If 11. The variation in a particular trait is normally distributed in a population before directional selection begins to operate, it might become markedly twisted in one direction afterwards. The variation in a particular trait is normally distributed in a population before directional selection begins to operate, it might become markedly twisted in one direction afterwards. Directional selection in its extreme form gradually eliminates recessive homozygous individuals from the population. Another type of selection is artificial selection, which is created by humans rather than managed by natural conditions. It is the process of enriching a population for a trait beneficial to human beings, which occurs far more rapidly under the artificial situation. However, S. Spiegelman and colleagues reported that a rigorous selection for one trait can lead to the drastic elimination of many other traits. 8.2. Mutation. Mutation is an evolutionary agent, and mutability is a required property of the genetic material if evolution has to occur. Mutation brings about evolutionary changes through several ways. Mutation may be highly directed at a particular locus such that allele A1 is selectively driven to the A2 form. Alternatively, the mutation might be random process, but with time, A2 form would come to predominate over A1. Finally, mutation might provide a population with new alleles on which any and all evolutionary agents, natural selection, for example, can act. In any particular case, mutation may progress only in one direction, as a right-pointing arrow A, or it may proceed in both directions A left-right arrow A. In first case, a slight mutation pressure may eventually leads to a species homozygous for the mutant gene, unless it is prevented by a selective disadvantage of mutant. In the later case, if all of the genotypes has equal selective value, then the frequencies of the two alleles would reach an equilibrium, the numerical value of which would depend upon the actual magnitude of the mutation rates in the two directions. The equilibrium point is related to the two mutation rates in a simple way. If rate a right-pointing arrow A equals U and a right-pointing arrow A equals the frequency of A at equilibrium. For example, if the rates are equal, then, or equilibrium will occur when the frequencies of A and R are equal. If U equals 2, then A is being formed twice as rapidly as A, and, or equilibrium will be reached when 2/3 of the genes are A. If U equals 4, then, 80% of the genes will be at equilibrium. 12. The sickle cell trait is also apropos here. The Hardy-Weinberg formula gave an expectation of 0.64 ss normal homozygous, 0.32 ss heterozygous, 0.04 ss sickle cell homozygous in African tribes studied. Random breeding should give normal and heterozygotes in a ratio of 2:1, or 0.67 to 0.33, with SS being subject to 100% negative selection. This should cause a drop of 7% in the frequency of S and a similar rise in S in one generation. Thus, S should be rapidly eliminated, however the population appears to be in equilibrium. This can be explained by the fact that selective elimination of SS individuals by malaria gives a selective advantage to the genotype S's, which confers resistance to malaria, in spite of the severe selection against homozygotes for S. In nature, mutation and selection always act simultaneously, perhaps in same direction or in opposite direction to upset the Hardy-Weinberg equilibrium. Most frequently, selection will work against mutation, as the majority of the mutations are deleterious. This will result in very slow changes, if any. But if a particular mutation is favored by selection, and if its mutation and selection might well cause a rather rapid change. 8.3. Meiotic drive and migration pressure. Besides directional selection, natural and artificial, and mutation pressure, meiotic drive and migration pressure are also responsible for shifting the gene frequencies in a population out of Hardy-Weinberg equilibrium. 1. Meiotic drive. The Hardy-Weinberg concept of an equilibrium population assumes that alleles will segregate at 1:1 ratio at meiosis and all gametes in the pool have equal chances of fertilizing one another. Hence, if gametes of genotype A1 have more success rate in fertilization than gametes of genotype A2, the